Thank you for participating in the NIA and NHLBI Entrepreneur Workshop Series. This workshop covers the topic of building your regulatory strategy. This breakout room, moderated by Dr. Chris Sasiela, discusses the topic of drugs and biologics.
Well, welcome and good afternoon to all of you. Thank you for joining us today. As mentioned earlier, my name is Chris Sasiela. I answer so much easier to Chris than any attempt to pronounce my last name with the word doctor in front of it. So please call me Chris. I am going to be video off during our presentation, and I may also end up, strangely, being audio off for a few moments because in the way of the world, there is a delivery that's going to arrive at my house while we are on the phone. I promise to stay longer to make sure I cover any and all time that gets taken up by that. I appreciate those people who did send questions in advance. The organizers, Ogilvy, did an excellent job of providing all of those questions to me if they were submitted in advance of this webinar, and I was able to take a look at them this morning. And I saw that they kind of landed in a couple of major buckets. I want to cover those buckets with you, though I may not address your specific question because there's a lot of repetition in the questions. So I am going to give you a heads up on the way that I broke these out, and then we'll kind of dive in. I will go in the order in which I have organized them, but feel free to interrupt for clarification anywhere along the way. So the first series of questions were about early interaction or appropriate timing for interaction with FDA, right? And so what I'm going to touch on during that discussion is going to be the pre-IND meeting process, the interact meeting process, and when and how you can get that very early informative information and how to find that. Some of it's on the website related to making sure that you have enough information for FDA to agree to a meeting with you, because they will not agree to a meeting if you don't have a substantial enough amount of information or the right information. I also have a couple of links that I am going to probably end up dropping into the chat. Let me see if I can do that right now. Actually, there are 2 webinars that are available online. These are talks, that I'm having trouble. These are talks that Ben and I gave last December. It was almost a year ago. Wow, that's been a long time. And in those webinars, trust me, the links work. They don't look like they're links, but they are. So just click on the name of either the drugs and biologics or the devices and diagnostics. And what we cover in those webinars is a really good overview of how to find information on the World Wide Web about your type of technology and how to prepare for that first interaction with FDA. So for drugs and biologics, that first interaction with FDA often comes in the form of walking up to someone when you're at a meeting and asking them, so I'm developing this product and I think it might be this. Can you tell me, because you're from FDA, what I should do? And that FDA person will very politely but firmly say, I'm sorry, I'm not going to talk to you. Or I'm not going to give you useful information anyway, because there is a process and they need to go through their process. So what you can do to find early information about the types of early animals that you should be using to test the potential efficacy of your drug, or to explore formulation options, to do some preliminary dose ranging studies. All of that can be found in guidance documents on the FDA website. And as Ben mentioned during his talk, the easiest way to search the FDA website is to use your favorite web browser. whether it's Bing or Google or Mozilla or whatever the heck it is, type in there something along the lines of dose range studies, Alzheimer's disease, right? Maybe you'll get a really good hit. Maybe you won't. If you put the acronym FDA in there, you'll probably get better hits than if you don't. If you are developing a drug or a therapy that is for an indication where there's already an approved use or an approved therapy on market, you can look at the approval documentation for that on market drug and see what they had to prove in order to get on market. What was the burden? How many patients did they have to enroll in their clinical trials? How many different animal studies did they do? What animal models did FDA accept for them? Right, all of that's online, and how to access all of that is in those webinars. So I want to start by just really encouraging you to pull that link down and make sure that you do take the time to look at that. During the earlier talk session, someone put in a question asking, what is an Interact meeting? They had not heard of that before. So the Interact meeting is a meeting that is very specific in the Center for Biologics. And this was started by what used to be the Office of Cell, Tissue, and Gene Therapy, OCTGT. It was a way for them, because they were working with products that had no precedence. And so each investigator that came along developing a new drug really needed very personalized interaction with them. There were no guidance documents. There weren't prior approval documents you could look at. So this was a way for them to say, here's a process. You're going to send us a certain amount of information about what you're developing, what stage you are at in that development, what information you have about it, why you think it might be effective. And we're going to talk about the work you will need to have in place in order to get a pre-IND meeting. So we'll help you identify animal models and talk about the pluses and minuses and whether we would find them acceptable. We'll talk about how to do dose ranging studies when what you have is something very specifically designed to touch humans, because perhaps it's an antibody or a fusion protein of some sort. And then it won't work in any other animal model because it's really targeting the human space, right? So they had those conversations in these Interact meetings. At the time, they were called pre-IND meetings. And they are now a formalized process across CBER. And some of the Center for Drug Evaluation and Research divisions and offices have adopted a similar type of meeting. They don't call it an Interact meeting there, but they do have a pre-IND meeting. These are things not for new developers, but for new technologies. FDA will grant you a meeting when they need to learn about what you're doing, or when you are asking for reasonable feedback on your development plan. You can't go to them and ask, how should I do this? But you can say, here's my plan. Here's the reason. There's a data packet behind every request. Here's the reason why I think this is the right approach for me to take. Do you agree with my approach? If FDA agrees with your approach, you're good. If they don't agree with your approach, they will fill in the difference. They will say, you proposed this. We don't believe that is sufficient because, and we'll give you paragraphs of that. Or you proposed this and we think that might be too much. We want you to scale that back. You shouldn't be putting your resources there right now. I've seen both sets of advice come back. When you are requesting a meeting, whether it's an Interact meeting, a pre-IND meeting, a pre-IND meeting, and a pre-IND meeting is the most common meeting for drug and biologics developers at NIH to request. You come in and there are 3 topics that you cover in that conversation. You cover your chemistry, manufacturing, and controls. how you're making what you're making, how you are validating its purity, identity, potency, etc. And then you cover, one moment, Abby, I see your hand is up, thank you. Then you cover the preclinical development plan. You're planning on testing this many animals in these sorts of ways to gather that type of data, which you'll present in your IND. And you cover potentially your phase 1 clinical plan, right? And both the development work you're doing and the preclinical data should support the first in human exposure that you're proposing in that first clinical plan. Abby.
Yes, I definitely didn't want to interrupt you because it doesn't really have to do with your explanation of the Interact meeting and the difference between pre-IND meeting, but I wanted to get in there before I forgot. So thanks, Chris. So my question might likely be answered in the link that you provided to the webinar you posted about, appropriate when to, interactions with the FDA, how to request, when to request, et cetera. But I guess it's kind of more experiential what I want to ask you. So, so how much is too much? So for example, if you get a response, for example, like from a pre-IND meeting from the FDA, but they think that you might have missed the mark on a few things and they encourage you to request another pre-IND meeting. But they provide you some resources and folks that you can email and reach out to, during the process. So is that really an open invitation to kind of exchange ideas, or are they more looking for like, another sort of big package formal sort of submission through the proper FDA channel, to go through that kind of formal response process?
I understand. So if FDA says to you, please come back with a second pre-IND meeting, they're probably anticipating that will be several years down the line. That you have to go back and do a sufficient amount of additional development work that they will then want to hear from you before you submit an IND. I'm sorry, I need to put you guys on mute for just one moment to let these people know that I am here.
Thanks, Chris.
No worries, Chris. Thank you. Thank you for the question, Abby.
While she's stepping away for a moment, I'll just give another plug to, if you could please open the feedback form. I'm going to paste it into the chat now. We'd love your feedback as we continue to have these events and really try to make them as beneficial as we can for our audience.
Agreed. Thank you very much, Lanny, and thank you to everyone for attending.
And thank you for your patience. I do apologize. I just knew that this was going to end up being the case. And I honestly did try to reschedule this.
Thank you.
So the first part of the question, if you'll just pull me back into the room here, Abby, we covered the requesting a second meeting and what the expectations might be around that. The second part of your question was, is this really a thing where between meetings I can interact with FDA? Yes, that is really a thing. And yes, you can become a pest.
Awesome.
Or you can use that relationship appropriately. When I've been working with companies on this for over a decade now. I did this before I came to NIH. I've been doing it the entire time I've been here. My rule of thumb is, if your product development is going to stop because you don't have a piece of information that only FDA can provide, then you need to be reaching out to FDA. Right? There are people who are developing products and like, oh, I'm completely unique. There's nothing like me in the world. And that is what NIH funds. So I hear that a lot. But it is often the case that although you are unique, you are still like something else. That something else may be that you're developing an eye drop as opposed to a tablet, where you are developing a tablet instead of something that is infused, right? But you're still like that other thing in some way. You're treating the same indication, you're targeting the same population, right? If that exists, then you want to do that research. You want to go find that approval documentation and see what exists already. Are there guidance documents out there that talk about what type of preclinical data FDA wants to see for your indication? Right? All the information you can find without going to FDA, they will be annoyed if you come and ask them to do that for you. That's not their job. They already did their job. They published it. It's in the public domain. It's your job to find it, which can be a challenge, but not something that you have to do on your own. That's part of what the seed office does, is that we have, as you've seen with Francisca and Ben, we have regulatory subject matter experts on staff, and we will help you understand how to find that information, how to start walking that path. But there will
Thank
be
you so
things,
much.
yeah, there will be things that no one else can answer, and you should absolutely reach out to that person at that time. And if there's something in their response, because anytime you communicate with them, they respond. If there's something you don't understand in their response, you can go back and ask for clarity. The word should means just that. You should do this. If you do it this way, there's a high probability FDA will be okay with what you're doing. But you don't have to. It doesn't say must, it says should. And so since you should do this, if you choose to do something else, you just need to justify it scientifically. And it needs to make sense to FDA, and they have to agree that it, whatever you do, is an appropriate alternate to what they proposed they're familiar with. So you can have those sorts of clarifying back and forth interactions also. Okay, how are we feeling about this concept of the early interactions and how they work? Do we have any additional questions on that, or shall we move on to the next topic? I see the question about where can I find the approval documentation? There are databases on the FDA website where you can pull up approval documentation. The small molecule drug approval documentation can be accessed at drugs@fda.com. The biologics approval documentation is a little trickier. There is a biologic approval documentation database. It's organized by year. It's not as easily searchable as the small molecule one, but biologics approves many fewer therapies than CDER, so it's not insurmountable. Once you go on, and if you watch the webinars, you'll see you can run through the series of clicks that will get you to where you want to go. Okay. The next set of questions that I saw in the pre-submitted questions talked about product development, preclinical development, route of administration, and formulation type questions. And those questions went along the lines of, I want to better understand the CMC requirements, or what sort of reagents I can use. When do I need to start being more GMP-like? GMP means good manufacturing practices. And how clean do I need to be? How clean does my product and my process need to be in order to get to first in human? How much information do I need to have about it? These are things that, depending on your indication, can be either very strict or very lax. If they are strict, that means you're probably going for a relatively large indication where you're going to be exposing the population, a large segment of the population, to whatever therapy it is you're developing. If the regulations are more lax, then you are likely looking at something that's treating a life-threatening disease, right? Or something that is rapidly progressing. And they're going to allow you to go in, this is how cancer drug development traditionally works. They will accept a much more relaxed version of GMP for a phase 1 oncology trial than they will for a phase 1 blood pressure trial. There are many effective therapies for blood pressure already. There are several effective therapies for cancer, but there's still much more of an unmet need in the cancer space. So those sorts of questions, asking about the parameters, the assay parameters for your certificate of analysis for your phase 1 material, is something that you want to do in that pre-IND meeting. So really the difference from FDA's perspective comes in the form of the severity of the disease. There was a question about the use of fetal bovine serum and other non-GMP grade materials. And this is a question in biologic development. At some point, you are highly likely to need to be in a serum-free media. Certainly, animal serum is a huge challenge, right? Because we have all sorts of prion-based diseases that can be transferred through the use of those materials. How far out from initial development of your master cell bank or your working cell bank you need to be when you start weaning that out and optimizing your growth conditions to a serum-free media is a conversation you can have with FDA. Like if you are 10 generations out at the time that you want to get to clinical trials, is that sufficient? Do you need to be 20 generations out? I don't know the answer. That will be a case-by-case basis. There was a question that talked about differences in route of administration. So most drugs are orally taken, or many are also injected, whether they're injected sub-Q, infused, whatever. But then there are also inhaled drugs, and then there are eye drops, and there are lotions, and there are unguents or oily type things like a Neosporin sort of solution. There is not necessarily a difference in the way that you would go about developing that drug. However, you will need to, in almost every case that I know of, you need to do your preclinical development using the same route of administration that you will be using when you are actually going into your human trials. So if you are planning an inhaled drug development program, then you need to make sure that you're doing your large animal IND enabling studies, and ideally earlier studies, using an inhaled formulation. Now, do you need to use the exact same nebulizer or propellant device in those studies? Possibly not, but if you can, that's much better. But you can't go from a swallowed formulation in your animal studies to an inhaled solution in your human trials. That data will not pass muster with FDA. Additional questions about route of administration? How does one deal with supply chain issues, especially if your product is midway through manufacturing? I'm not a supply chain expert. There are all sorts of things, unfortunately, that are really impacted by the supply chain issues that have been exacerbated by the global pandemic. One way to deal with things like that is to ensure that you have a plentiful and varied number of potential vendors for as many of your raw materials as you can manage, right? So if you are using salt and sugar and you're creating your own ultra distilled water on site, right? But you're buying all of the reagents to make all of your buffers, and you're buying the lactose and the hydroxymethylcellulose and the whatever else it is. You want to have multiple qualified vendors. That is your best security against supply chain issues. That's the only security I know against supply chain issues. And in a scenario like we're in today, even those protections are just not going to be sufficient to cover the reality of a global shortage of many, many things. Okay, I see Chris posted a, oh, we're running faster into the questions than we're going to have time for. I will stay though. I will stay for as long as I can. Next, recently added question: when is it recommended to have a pre-IND meeting with the FDA? So I will give you the answer I have given for a decade or more. You should meet with FDA before you spend $500,000 or something close to it on your IND enabling large animal studies. You should meet with FDA before you build a manufacturing facility or commit to developing and manufacturing your phase 1 material. Ideally, you should meet with them before you manufacture your IND enabling study material. Only in that way can you be sure that the data you're about to collect will support your IND. If you have not met with them by then, you are late to the game. You don't want to be late to the game. You want to be streamlined, slick, fast. Okay, hopefully that helped. David posted DMF specifies reagents and qualifications, validation in great detail. Was that a comment for the room or was that a question, David?
Comment response to the discussion a couple lines back.
Ah, great. Thank you, David. And David is absolutely correct. Your DMF, your drug master file is a huge resource for you. And you can learn more about what all of that entails on another day. Chris has asked, what about if you want to change the route of administration of an approved drug? For example, from oral to IV. How much preclinical is needed? Well, if you are taking an improved drug and you're developing it for the exact same indication in the exact same population, then a relatively small study is probably going to be required. If, however, you are looking to take an approved drug and move it into a new indication and a new population, which is much more likely to be the case with NIH-funded research, then you are going to need to propose a reasonably substantive set of data when you go to FDA. Now, you'll be able to bypass some of the early safety studies, right? Because that's just what it's going to be. It's going to be a 505(b)(2) most likely. And that is a slightly abbreviated path. You will be able to rely on existing safety data, existing published data, but you're still going to have to do some work. How much existing data there is in part going to be how long has the drug been on market? How different is the indication that you're looking for from the indication it's currently approved for? And is there anything that is inherently done when the drug passes through the gut for an ingested drug that would not happen? Is there an important first pass metabolism impact for that drug that allows it to be active or less active so that the tolerance and the therapeutic index might change? You would need to investigate those things when looking to change your route of administration. Okay. What is the timeline between pre-IND meeting and interact meeting. So the timeline between any meetings at FDA is always driven by your development work. When I was at FDA, when I was receiving my initial training in regulatory, we would have very experienced companies coming in, and they would have their pre-IND meeting effectively 6 months before their IND filing, because this was drug number NNN that they were developing. They knew what to do. They had really good teams, and they didn't need to have those meetings quite as frequently. And they just wanted to make sure everything was in place, that their IND would be accepted, or that it wouldn't be accepted, but they still wanted to meet their year-end goal of filing it, which was one of the more confusing meetings that I attended. For most NIH innovators, it's how long does it take you to run through the information you gained from FDA during that prior meeting. If your interact meeting set you up for 5 years worth of work and you're getting the results you expected and you're still following the guidance you got from FDA, then it's 5 years until your pre-IND meeting. On the other hand, if you are following that guidance and you're not getting the results and you need to change approaches, request a meeting sooner. If you're following that guidance and it's all going swimmingly and you only have 2 years between your interact meeting and your pre-IND meeting, that's cool too. FDA doesn't set that timeline, you do. The science does. And in many cases, the funding does, unfortunately. The timing of pre-IND request, what do we need to deliver to get an answer, and how long till FDA grants a meeting written response? There is actually a guidance document that I am going to drop a link to. I'll send the link actually to Lainey and the Ogilvy organizers to send out to you guys. Or you can look up, the guidance document is called Formal Meetings Between FDA and PDUFA Sponsors. So in that, it describes the 3 primary types of meetings, A, B, and C. Pre-IND meetings are a type B meeting. You need to submit a cover letter that has your questions broken out by discipline. Behind that cover letter is all the information you have that FDA will use to support their analysis and create their response to your questions. If you do not give them enough information to respond, they cannot give you a viable response. They can't do that. The time from submitting that request to having the meeting is technically 60-ish days. You get to tell them when your team is available. They put together their team and look at calendars and they respond with something that works. When you put in a pre-IND meeting request, if it is granted, you will always get a written response from FDA in advance of the day of the meeting. Now, that written response will be thorough, it will be complete, it will tell you everything you ever wanted to know, and you will be completely confused by it. Any place where they say, yes, your plan is good, you can say, phew. Anytime they say, no, your plan is not appropriate for what you're doing, then you need to take a hard look at those no responses and what they propose you should be doing instead, and decide if you want to continue and move forward with having the meeting. If you want to continue and move forward with having the meeting, they will all get on the phone. There are no in-person meetings at this point, but they will get on the phone with you, and you will have 1 hour in which to discuss those areas where you and they do not agree on your development plan. You'll ask them for clarification. You might propose, like do some sort of negotiating. We see you're requesting this. Is that absolutely a requirement or is that a suggestion? One of the things most people don't know is that you can say, thank you very much, we appreciate your input, and then not do the thing. I have been in meetings where FDA said, here are the things that would put you on hold if you were to submit an IND, and here are things we would like to see also. You'll always get those. Those things they would also like to see, you can say, that's great, we'll think about that later in development. We don't need to focus there in order to get to first-in-human trials. It's always a good plan. I would put it as mandatory actually, but I can't because that's not my place. To have a qualified regulatory professional on your team when you are having a pre-IND meeting or an interact meeting. You really want someone who's been down that road, had.
Chris seems to have audio issues.
Yeah, look, she's messaging me. Looks like she's having just a little bit of trouble.
And I saw that we were, I think we had one more question to go. And if we can't get their questions live, we will follow up with the answers. Maybe Chris, could you answer the last question or two in the chat? And then everyone can get their questions in real time. Thank you, Chris. So in terms of some of the answers that she came in, one was letting people know that the CBER pre-IND was an informal process and it was changed to interact about 8 to 10 years ago when it was formalized. She also clarified for the question about the GMP facility that if your pre-IND meeting was rejected for lack of ability to reproducibly manufacture your drug product, you do need to prove that you can do that, whether it's in the CGMP facility or not. And then there's a question about the FDA Biomarker Qualification program. And she didn't know too much about that. That might have been covered in one of the other breakouts, but information should be available online. Dr. Parsons, yeah, I'm not sure. I mean, you'd have to go to the FDA why they cannot discuss it or what the lack of clarity is there. And Chris, I haven't seen the answer. Could you say if the interact is specific to biologics? Okay, it is specific to biologics. That's why we have Chris. To CBER at least, which covers biologics. Okay, well, I sorry for the audio issues here, but I think we may one more question. Okay, this is gonna be the last question for Chris, and it's drugs developed with stem cell models and data derived good for FDA. And this is I mean, I know from my own experience talking to FDA about this, I mean, you know, it's something they're actively looking at. I'm not sure what guidance has come recently, if any, but you know, an area that's obviously growing in terms of what qualifies as validated models, and I think something that will continue to be in flux as more of these models undergo additional validation. So it probably depends on the specific model, but I'll yield to Chris on any additional answers there. Typing the answer, I'll just thank everyone again, and please remember the feedback form. And thank you all for joining. And sorry about the audio issues. Thank you, Chris, for helping us answer those final questions, even when your audio is not working. Yeah, so I mean, I think, yeah, she said she needs more info. I mean, I think it definitely depends on the model. I know, you know, I worked on a funding opportunity around 3D models, in vitro models, a few years back, and at the time, you know, FDA was starting to incorporate some of them and consider the incorporation of some of that data into requests. I think it really depends on the specifics of the model and what validation has been done. But, you know, you'll definitely have to discuss that with FDA. So on that, we will close. And I think one of the other ones led by Ben Berman is going till 4:30. So if you have additional questions that you think pertain to that session, then I do encourage you to go to that session.
You can
go ahead, Lainey.
Yep, I was just gonna say you can do that by leaving the room, but not the meeting. And then once you're in the main session, we can help direct you to the open breakout room. But thank you everyone very much for joining and having an interactive discussion. We hope you found the workshop beneficial.